Welcome back. So, as you'll remember, we're in the middle of actually a very long series of vignettes, all of which are aimed at trying to show the use of rate equations and the use of rate equations to find the time evolution of interesting biological systems. And I'm trying to give you a potpourri, actually, of examples. So we've talked about ligand receptor binding, we've talked about enzymes and compartmentalization of enzymes. I'm going to talk about the SIR model, which is relevant to pandemics. And for the next couple of vignettes, I want to talk about gene regulatory networks, which will also serve as a very nice proving ground for these ideas. So this very brief vignette is intended to introduce the idea of gene regulatory networks from bacteria to humans. And the idea is really, I would say, relatively simple, at least in conception. And that is that if you take the E. coli genome, so, it's first of all, it's a circle of around 5 megabase pairs. So I'm just making a schematic of the E. coli genome. And this genome, the way that I would think of it, and I think we talked about this earlier in the course, was if I have roughly 1,000 base pairs per gene, and the genome, so the number of genes is going to be estimated, at least in the case of a bacterium, it's going to be estimated as genome length divided by length per gene, which in our case then will be something like 5 times 10 to the 6 divided by 10 to the 3 per gene, which gives me on the order of 5,000 genes. That's an overestimate, but that's the essence of the idea. Now, the idea of gene regulatory networks traces back, for starters, to Jacob and Monod, and then in the middle 1960s, people realized that the repression architecture was not the only architecture. That's a very interesting story because in the early 1960s, there were many universalities being discovered in biochemistry, which is really very compelling. So every organism seemed to share the same metabolic pathways. Every organism had a membrane potential. Every organism had the same genetic code. They all used ATP. I'm exaggerating a little bit, making this too strong. With biology's full of exceptions, but the point is that there's all this sort of, let me call it universality, and the intriguing thought that Jacob and Monod had was, okay, well, maybe in the context of regulation, all regulation will be the same. Already it was a discovery, the fact that there is regulation, but once they made that discovery and they attributed it to repression, they thought, well, maybe all regulatory interventions are going to be of the repressor variety. That turned out not to be true, and it was a hard-won battle. And interesting, Jacob and Monod went from being the revolutionaries to being the old guard. And what I mean by that is that, they basically were kind of resistant to this idea of activation. And it took a 10-year battle to work that out. But what I would say is in the late 1960s, people like Britton and Eric Davidson posited that genes were connected in more sophisticated networks where the output of one gene ends up controlling another, and maybe a given gene has combinatorial control, so you have multiple inputs into that gene and so on. And so that now, in the full maturity 50 years later, has led to a picture of genes that are connected in gene regulatory networks. And what I want to show here is, first of all, kind of a schematic of what one might mean by that. This is from Uri Alon's book. We repurposed it, but what I want you to imagine here is that you have these transcription factors, X1, X2, X3, XM, and each one of those transcription factors may respond to one or several signals, and we're going to talk later in the course about what we even mean for a transcription factor to respond to a signal. That's going to take us to what I like to call biology's greatest model and thinking about allostery. So very fun and interesting stuff. And then what you see is that these transcription factors make interventions on a variety of genes. So for example, we see that X1, the transcription factor X1 interacts with gene 1, gene 2, gene 3, and gene 5, and so on and so forth. And when I say interact with, what I mean is that to first order, I mean either they repress or they activate those genes. And for the moment, I'll leave that kind of vague. So that's what I mean by a gene regulatory network. It's the wiring diagram that tells you how batteries of genes are connected to each other in the sense that the output of one gene ends up being the input to another. And this is an example from E. coli. And what I want you to see, for example, over on the right-hand side, you can see that CRP is a big node. And when I say node, what I mean by that is that it's a master regulator. It interacts with a lot of different stuff. And you can see other examples where they interact quite modestly. In other words, they don't have very many targets. ARO-C is an interesting one that maybe we'll come back to later. LAC-I is for sure an interesting one that we'll talk about. And you can see both of those don't really talk to that many genes. So LAC-I in particular controls LAC-Z, LAC-Y, maybe LAC-A, I can't quite remember. But anyway, so this is a gene regulatory network in the context of E. coli. Another one that I like is PureR, having to do with purine biosynthesis, and I'll probably say something about that as well later. So I also wanted to just give you an indication. This is from Rick Young at MIT. This is a series of discoveries that they made in the early 2000s. This is now in the context of yeast, and I think this is a useful one for our purposes because it gives you a sense of some of the different regulatory motifs that you would find. And what I mean by that, let's just review them. So the one we're going to talk about in the next vignette is shown at the top, which is autoactivation. The idea is that the gene product, in that case STE12, actually then goes back and it activates its own gene. So, as I show you here, this guy actually comes back and binds on its own gene and activates it. So that's autoactivation. Mutual activation, that's a cool one, because that has the output of one activate another, and then that other one goes back and activates the first. So mutual activation is a really nice motif that we'll think about. Then a very ubiquitous one that Uri Alon has made a big deal about, I think, which is cool, is the feed-forward loop, which you see here, which is gene 1 activates gene 2 activates gene 3, but also gene 1 activates gene 3. And this thing has interesting dynamical properties. Simple activation is what it sounds like. It's just that there's some transcription factor, which is shown here, and it activates various genes. Combinatorial control refers to the fact that there are many genes, and this is even true in E. coli, which have multiple inputs. So in other words, there's not just one transcriptional input, but there's multiple. And together, that gives rise to the possibility of logic. What I mean by that is, you have 2 to the n possible decorations of that promoter with transcription factors. And in principle, each one of those different inputs could have a different output. So that gives you this really wide berth for thinking about regulatory combinatorial control. So that's my introduction. This is just to, in a way, serve as a preface to what's going to come now. And the connection to the dynamics part of the course is we're going to now ask ourselves questions such as, what is the time rate of change of the gene product number 1, and that might be a function of gene 2 and gene 3, etc. In other words, these other genes might be inputs. And so we want to write down the rate equations that describe the time evolution of these systems. And so that's what's going to occupy our minds for the next couple of vignettes, where what we'll think about is switches, first of all, and we'll consider the mutual activation switch and then the mutual repression switch. And then after that, we'll think about the double negative architecture, and we will think about oscillators. So that'll give us a lot of opportunity to explore it's sort of a dual exploration that's useful on several fronts. One is it will be very instructive in thinking about gene regulation itself, and that in itself is a useful thing for us to contemplate from a biophysical perspective. But also it will give us an opportunity to continue to expand our toolkit and our sort of felicity with the application of these rate equation approaches. So that's what we have on tap.